aim of the project is to develop a sterilizable endoscopic camera calibration device that can be used in operating theaters by clinicians.
Medical imaging methods, such as real-time mosaicing of the human placenta vasculature, currently suffer from optical distortion effect that could be mitigated by a standardized calibration process.
This device can serve as a quick and practical tool for the peri-operative calibration of endoscopes used in fetal surgery procedures, such as twin-to-twin transfusion syndrome.
Before sandblasting, use a ruler to draw a 40 by 40 mm square onto a 1.2 mm thick 316 stainless steel sheet. Then, use a manual metal cutter to cut out the square and use a file to round the corners and sides of the sample. Next, place the steel onto a 1 to 2 cm thick block that is slightly larger than the square sample. Use one vise to secure the sample onto the block and another to hold the sample tightly by the edge of the block during the sanding. Then, move the entire assembly into an internal blast chamber. Turn on the compressed airflow and tightly seal the chamber. Then, don a pair of safety goggles and position a blast gun perpendicular to and at least 4 to 5 centimeters away from the metal surface. Next, apply the foot control to sandblast the sample. Then, sandblast the other side as just demonstrated as appropriate. For laser patterning of the sample, first design a pattern of asymmetric circles and use the appropriate design software to prepare a drawing exchange format file.